Again, journals start, some die, some thrive. This became one of the most important journals in medicine, and the summary is 10 lines. I want to read them because I believe in good science from the past, although it's not on Medline sometimes, and it's not as accessible to young people. So what Klaus Rogmann is a German who moved to Canada and spent most of his life there at the University of Manitoba, and Pena is one of his co-workers, but the real hero here is Klaus. What it says, the title is Mitochondrial Calcium Overload, a General Mechanism for Cell Necrosis in Muscle Diseases. And what it says is, it is suggested that the mechanism of muscle cell necrosis in various muscle diseases, so it's a general theory, is explained by an increased net influx of calcium into cells, which triggers a vicious cycle of mitochondrial calcium overloading and energy depletion. If correct, this hypothesis may offer the basis for a more rational treatment of some muscle diseases even before their primary etiology is known. And believe me, this is a very important paper. I'll show you the scheme they proposed. So it's general, deficiency of vitamin E, selenium, biotin, all kinds of things, abnormalities in muscular dystrophies, infective agents, immunology, whatever, would converge on the sarcolemma, cause this increased net influx of calcium, excess uptake of calcium by mitochondria, and then there is another keyword here. Functional and later structural damage of mitochondria. Dystrophic kid is normal until he becomes dystrophic. This means dystrophy is not there all the time. That means there are compensatory mechanisms that prevent disease from becoming a disease. So the natural history of disease tells us, yes, there is a functional compensation until that fails. So by studying these mechanisms, we can perhaps understand what goes wrong in the end. But this is very, very perceptive. And he made always a point, even dystrophic mouse models, they bite and go after you before they become dystrophic. That's true of the children as well. So eventually it's a crisis, but once lack of energy sets in, then the system enters the vicious cycle, less calcium is pumped out of the cell, and you can see that eventually cytoplasmic levels of calcium increase, giving hypercontracture of muscle fibers and cell necrosis. There is evidence that this might be the case, but this is 76. This is before FURA2, before it could be measured, actually. So I think this is absolutely outstanding. And what I'm going to discuss is genetic abnormalities in muscular dystrophies. Is this true? I got into this through the extracellular matrix because there was a model available in Padova created by Paolo Bonaldo of a collagen 6 deficient mouse. Collagen 6 is outside the sarcolemma, and we know that mutations are linked to two muscular dystrophies, Bethel myopathy and Ulrich congenital muscular dystrophy. And while I was working models of disease in muscle, Paolo came to my office and showed me these two electron microscopy slides. And you can see that some fibers in the collagen 6 deficient mouse had dilations of the sarcoplasmic reticulum and enlarged mitochondria. Not all of them, but this is normal, right? The mouse is still okay until it gets worse, and not all the fibers are affected. If you look at one interesting detail, though, we noticed immediately, or he noticed immediately, that the fibers with these abnormalities in the sarcolemma, in the SR and mitochondria, also had apoptotic nuclei. So is this a coincidence? How can you prove a mechanistic relationship? It's not easy on an image, right? So what we noticed are swollen mitochondria, and the question was, is this the consequence? Or could this be part of the cause of the disease? So we spent 10 years working at this. I will just give you the punchline, because I want to give you some interesting data we have just published. So in the end, we showed that, yes, mitochondrial dysfunction does play a causal role in the pathogenesis. And this is due to inappropriate opening of a high conductance channel in the inner membrane called the permeability transition pore. This is the idea. Inner membranes can open this pore for long times. It's a big channel that allows solute diffusion and then water, and then mitochondria swell. And they lose cytochrome C and NAD. We heard an interesting story about NAD in mitochondria yesterday, and I will tell you why it's important in this context as well. So one consequence is also depletion of NAD. Today we know that the pore can also open for short times, flickering. This is probably part of the physiology, but one can get into the other. We have many ways of controlling it. For today's sake, remember NAD being lost, and the only pharmacological tool we had so far was cyclosporin A. And I'll tell you how cyclosporin A enters into the picture now. Cyclosporin A has a receptor in the matrix, cyclophilin D. You can see here cyclosporin binding to cyclophilin D, and cyclophilin D is the mitochondrial target of cyclosporin. This is a soluble protein, while I told you the transition pore is a big channel. So what we learned, it was a bit disappointing, is that cyclophilin D regulates this pore indirectly by binding and unbinding to it. Interestingly, cyclophilin itself can be targeted by signaling molecules. It can be phosphorylated, dephosphorylated, acetylated, S-nitrosylated, and this will in turn affect the propensity to interact with the pore and modulating it. So we used cyclosporin to test this idea that the pore was involved in the pathology, and it worked like magic. And within a week of treatment, the mice became, quotes, normal. I won't show you the functional data, but it's all there. Just a glimpse, I think that the pore is, in fact, a special conformation of the ATP synthase, which turns from the energy-conserving machine into a pore. And I will not tell you much about this today, other than with inhibitors you can prevent this from happening. I will tell you also that this topic is very controversial, and if you want to read the latest, I have a very brief comment that accounts for all current discrepancies, apparent discrepancies and controversies. So I won't go into this at all. I have this list just to show you that I spent Telethon money fruitfully. It took us a long time. Perhaps the most striking proof of concept that cyclophilin D is involved in this pathology is that if you cross cyclophilin D null mice that have no receptor with the collagen 6 mice, you get a repair of the disease. In other words, you're removing the target inside the mitochondria, and you're fixing a problem that starts at the extracellular matrix. And because of this, we were allowed to do a pilot trial on 5 patients that gave very encouraging initial results. Unfortunately, the long-term effects were not so brilliant. In particular, the patients did not recover from diaphragm insufficiency, and in fact, that kept going on as if they were not treated. Why is this? Now, I will tell you a great experience I had in pharmacology. Of course, cyclosporin has multiple effects, and you all know it as an immunosuppressant. So I just mentioned cyclophilin D inside mitochondria as its receptor, but of course we have a cyclophilin A in the cytosol, cyclophilin B in the ER, and cyclosporin blocks them all. So the first issue is cyclosporin selective for the mitochondrial effect? And the answer is no, of course not. But what's more important is that the complex of cyclosporin and cyclophilin will inhibit calcineurin, and this prevents the translocation of NFAT to the nucleus. And this is the basis for the immune suppressive effect. There's a second important effect of calcineurin, dephosphorylation of the RP1, and this is also inhibited by cyclosporin. And this has mitochondrial effects that have nothing to do with my pore. So there are many reasons why the effects could fail or why you can get toxicity. But the key point was, it was not clear at all whether inhibition of calcineurin is good, bad, or neutral. Why? Because the mice are not too sick. And since you can cure them with cyclosporin, you can't tell whether something else is better or worse, because you can't separate inhibition of calcineurin. And then the second thing is that the mouse is not very sick. We've seen this yesterday for the MDX mice. Same thing here. Sometimes you cure the disease because it's very mild or because the mouse has compensatory mechanisms that are not there in men. So it was not enough. Luckily, cyclosporin is a well-studied molecule. You can divide it into two functional domains. This domain here is the side that binds cyclophilins. You've seen it in my picture, face on. Once that happens, this new molecule interacts with calcineurin with this free side. I have a movie, but it won't work, so we'll skip it. But to make a long story short, the chemists at Sandoz, now it's called Novartis, and also at the University of Lausanne, by playing with these two residues, 3 and 4, were able to produce molecules that still bind cyclophilins, but do not bind calcineurin anymore. In other words, we have a drug now that only hits cyclophilins without the calcineurin side effect. But how, this is the new model, zebrafish. This was for you, Giovanni. Isn't it funny? Do you believe this is true? A fish that looks like a zebra? No, but it's called zebrafish because it has nice stripes. So this is the real zebrafish. And it's possible to reproduce many human diseases in zebrafish. And in fact, Marco Schiavone, who is sitting right there in our lab, was able to create these collagen 6 knockdown animals. I'll skip all the details. There is no protein there. They're dystrophic. But what's really important is that they're almost paralyzed. This is coiling. Normal fish move around in your dish. These guys are basically paralyzed because of the muscle problems they have. And so now it's the ideal system because it's so severe that we can compare cyclosporin, which inhibits both cyclophilins and calcineurin. We can compare it with FK506, which only inhibits calcineurin, has no mitochondrial effects. With this derivative, NIM811, which is one of those who don't inhibit calcineurin and only inhibit cyclophilins. Now we can have the comparison. So cyclosporin did something, not much. FK did nothing. We start from here, right? And when we saw the NIM data, I couldn't believe my eyes. Full recovery. So lesson number 1, if you inhibit calcineurin, you're going to get damage. If you inhibit cyclophilin, that's good. So now we know that you should not inhibit calcineurin, and now we can use this in severe models. Just to show you how sick the animals look, this is the myotendinous junction. You see herniations through it, enlarged mitochondria, herniation of the outer membrane. These are too large compared to normal. And after the treatment, you cannot tell them apart from the wild type. So it's really impressive when it works. So this was the story of the fish. But the key question is this a general mechanism, as Klaus Rogman suggested, or this is just an oddity of this specific rare disease? And the most challenging thing we could do is try whether or not this also works in Duchenne muscular dystrophy. There were clues that this might have been the case. This is the work of the lab of Jeff Molkentin. And he investigated sarcoglycan deficient mice and LAMA2 deficient mice, another model of congenital muscular dystrophy, and the MDX mice. And he tried another non-immune suppressive derivative called Debio25 or alisporivir. I will use the two because they changed the name en route. But once again, the MDX mouse is not very sick. And the question was, will this work in a severe model of muscular dystrophy? And again, we resorted to fish. After checking back with Patrizia Sabatelli. This is a group Luciano Merlini and Nadir Maraldi. They're kind of old now, but they worked at the Rizzoli Hospital in Bologna. And she went on she had biopsies from patients, and it's so common to see strange mitochondria, swollen mitochondria. It's just anecdotal, if you wish, but it was interesting to find something that might have been overlooked in the real patients. Anyhow, we took cells from the patients, and this is respiration. So this respiration is how much oxygen is being used by the patient's cells. I draw a red line, and you see these are 3 patients. This is a healthy donor. They don't look very different from me and you. But this is basal respiration. If you're sitting and not doing any work, you may have enough to survive. But the question is, try and exercise, do some work, and let's see how fast you can run, how much oxygen can you use. And then a striking difference emerged. Now the green line is what a normal cell can do. You can go 2.5 times faster. The patients can't. So this means they have a very small respiratory reserve. Okay? These are intact cells, they look normal, but now we know that the potential to perform work is less. So the idea is, what if this is due to flickering of the pore? They're losing pyridine nucleotides, and that's why they can't go faster than that. So we treated them with this drug, alisporivir, nothing happened. The problem was we only treated them for an hour. When you lose an AD, you have to resynthesize it from scratch. You can't import it, and that takes time. So we tried and treated them for a week. And after a week, you cannot distinguish them from the controls. In other words, we have restored the respiratory capacity by inhibiting the pore for enough, a time that's long enough, 1 week. So time is a factor. But how about the real world? Again, we went to fish. This is a fish generated in Tübingen by random mutagenesis. You can tell from the birefringence, it looks pretty sick. The fibers are not refracting well because they are not regularly spaced. They turned out to have a nonsense mutation in exon 4 of the DMD gene, which luckily in the zebrafish is not on the X chromosome. It's autosomic. So you can breed them, and they look pretty sick actually. So this is called the sapie zebrafish. You can see detachment of the fibers from the myotendinous junction, and they hypercontract because they are not holding on to the junction, and they hypercontract easily. And the mitochondria are swollen, and the fibers are disorganized. And this is after a few days with this inhibitor of cyclophilin. You cannot tell them apart from the wild type. Remember, they don't have dystrophin. They don't have dystrophin. But we are fixing something wrong with the mitochondria, which does not depend directly on the lack of dystrophin, but must have been elicited by something. And we think the something is old Klaus Rogman's calcium idea. I'll show you just the last experiment, but it's very dramatic. So Marco did the respiration of the living animal. So we put it into, it's a fantastic system. You can measure respiration. So this is the wild type, and this is the poor sapie. It's less than 1/3. It's still alive. And this is alisporivir versus cyclosporin. No effect of cyclosporin, and alisporivir recovers almost entirely the respiration. Now, survival, cyclosporin kills them. And I'll tell you in a minute why it kills them. Alisporivir allows a better survival. It's not the ideal drug, but remember, these fish don't have dystrophin, but you're doubling their lifetime. Think in terms of children. If we are lucky, we might get somewhere. But my take-home message is different. Your patient doesn't walk that far, right? Is that because the muscle is gone, or is that because respiration is limiting? In other words, I don't think that all of it can be accounted for just by muscle defects. If there is a respiratory component, we might be able to tell very early. That's my biggest hope. If I'm right about this, then the patients should benefit very quickly. If part of the problem is functional, not structural. That's the bet. And the paper just came out. Teleton is trying to help us promoting a clinical trial. Not easy, there's a lot of competition, but this could be given to the patients way before they get into the prednisone. So this has been tested clinically, it's extremely effective as an inhibitor, and it's not toxic. There are thousands of controls treated for another. The reason why we have so many data is too long to explain. It was developed for HCV, actually. That's why it's called alisporivir. And then they decided to drop out, and we got the drug back, but we know it's pretty safe. I will conclude with an interesting observation. Also, the group in Padova here, coordinated by Elena Pegoraro, gets credit for this wonderful thing. So, some patients do better than others, and it's not clear why some patients have something genetic, it's in the families, and they walk for longer. So they time to wheelchair. And it was discovered that this is due to a genetic polymorphism in the actinin 3 gene. So these patients have a second mutation in the actinin gene, so they are weaker. But they walk longer, and this is because their fibers go from fast to slow. So the performance is worse, but the survival of the muscle is longer. It's a trade-off. What triggers the change? Calcineurin. The actinin 3 deficiency turns on calcineurin. Calcineurin makes them from fast to slow, and slow fibers are more resistant to dystrophy than fast fibers. So again, don't use cyclosporin. It's going to make it worse. Use the pure inhibitors, then you have a chance. Took us a long time to get there, but now we hope it's moving fast. And the interesting thing is that the sapia zebrafish has the same features as humans. This is the mitochondria, this is fast, and this is slow. Only the mitochondria in the fast fibers are affected. So we think there is hope because we are reproducing even the details of human muscular dystrophy in the fish, and the drug works. So let me go back to the scheme I started from. Functional, I think, is pore flickering, and later structural, it's pore opening full-blown and releasing factors and stopping making ATP. And this sets in the vicious circle. So if you go from transient to prolonged, you start the vicious cycle of ATP depletion, calcium deregulation that involves the SR and the fiber as a whole. But today we have many ways of intervening. We have, as I mentioned, alisporivir, but also we developed new inhibitors, dynamic anylides, benzamides, isoxazoles, which are being developed under the support of the Fondation Leduc. Many people contributed to this work. I don't want to read the list. I mentioned a few, but it's really going on for 25 years. And I want to acknowledge the funding agencies, in particular, the Teleton Foundation, 25 years of funding without interruption, by far my number one financing agency in this endeavor. But I have to say, Debiopharm, the producers of alisporivir, at some stage, they funded the research. They really wanted to see this into the clinic. Now they're kind of faltering. I don't understand well why. And Roche, because they own NIM 811, and when I switched to NIM 811 for reasons that are hard to I can't explain now, they also supported financially this research. Well, this is some of the people who did this work, just to give you an idea of the number of people and young people enthusiasm that still surrounds this area of research. And with this, I'll stop, and thank you for your attention, and thank you again, Ugo, for having me here today.